unicellular or acellular or multicellular. Anyway, the protoplasm that is further divided into two components. One is cytoplasm, second one is the nucleus. Third one is the cell skeletal protein that also provide rigidity to the cell. This nucleus again in itself, that is a double membrane perforated structure. It is the only structure of the cell whose membrane is discontinuous. अगर आप देखें तो then nucleus, that is nuclear envelope and nucleoplasm. If a nuclear envelope, that is the nuclear membrane. Second is nuclear membrane, again outer, inner and perinuclear space. Nuclear as a pore and nuclear pore complex. Nuclear as a pore has that varies from cell to cell, however 5 micron to more, and allow the passage for inlet and outlet of the substances, including input of and flux of nucleotides from cytosol to the nucleus, the nucleoplasm. Second one, nucleus synthesizes all kinds of the RNA, and this RNA leave the nucleus for cytoplasm for further as of functions like protein synthesis. And also the ribosomal RNA that constitute the ribosome, that is associated either with the membranes or it exists freely or also present in some kinds of organelle. Nuclear pore complex, another has important protein that maintains the nuclear as a pore or the periphery of the inner nuclear membrane, that has a specific protein that is known as lamine. And lamine has a protein that joins the chromosomal telomere to the nuclear membrane. At the time of cell reproduction, particularly as whenever prophase is going on, so then there is phosphorylations of the lamine protein, and phosphorylations of the lamine protein that causes disassembly of the nuclear membrane. Later one, it makes the continuity in between nucleoplasm and protoplasm for further assembly of the chromosome. So that one kind of protein, that is the lamine. Nucleoplasm, that is again nucleosome and chromatin. Nucleosome or cytosol that differs from cytoplasm and nucleoplasm in the sense that it has to contain only fluid environment. It does not also hold any membrane bound organelle or membrane bound as a component. That is either in both cases, cytoplasm, that is the cytosol, in case of nucleoplasm, that is the nucleosome, and also chromatin. This nucleosome contains nucleotides. There is two kinds of nucleotides, deoxynucleotides and ribonucleotides. One objective to be point here, deoxynucleotides or ribonucleotide. The ratio of the deoxyribonucleotide and ribonucleotide is always 2:1. So that concentration of ribonucleotide is much higher than that of the deoxyribonucleotides. One question is more, at the time of the DNA replications, whenever cell enters from G1 to S, at the end of G1, the concentration of deoxynucleotide increases more sharply. That is indicate that the cell has all set for DNA replication and ready for cell reproduction. So that concentration of deoxynucleotide that increases only during that period whenever a cell leave the G1 for the S phase. Ribonucleotide that is used only for synthesis of RNA, that may be several kinds of the RNA, genetic as well as non-genetic, etc. Second, water and inorganic substances maximum contain phosphate. These are phosphate because the ribonucleotide that exists in nucleoplasm in form of ATP, GTP, CTP, UTP. That is at the time of the transcriptions, this is ATP hydrolyzes or GTP or UTP or CTP hydrolyzes for these polymerizations. Then it leaves more phosphorus atom in organic phosphate, that is as a PO4. In spite of this increase in phosphate, the nucleus maintains its pH nearly about 8. Some books have written 8.5 or more than it maintained alkaline, even though it contains more phosphate. Because DNA replications in case of eukaryotes that happens in alkaline as a pH. Second as a chromatin, that is the nucleic acid, and as a protein. Nucleic acid, DNA or RNA, some amounts of, because RNA is all the times remain attached with that DNA, because transcription is going on for the protein synthesis or synthesis of other kinds of RNA like as a ribosomal RNA and as a transfer RNA, or other kinds microRNA, siRNA, interference RNA. All these kinds of RNA are synthesized during in the cell all these times, so that RNA always remain attached. The protein, associated protein and non-associated protein. Associated as a protein, that is histone protein. Non-associated as a protein, that mainly includes some as a proteins that stabilizes the nucleosome structure like nucleoplasmin. However, other proteins that remain present in the nucleoplasm, that may be, that is RNA polymerase, DNA polymerase, and RNA polymerase is of in 3 kinds: RNA pole first, RNA pole second, and RNA pole third. But as a DNA polymerase, again, one point is noted: the concentration of DNA polymerase, particularly as a DNA pole alpha, beta, gamma, delta, or epsilon. The DNA pole as a delta, that is essential for DNA replication in case of eukaryotes. Its concentration rises from cell in the cell from when as a cell enters from G1 to S. Again, it might indicate the cell has all set. When I add, unless and until if DNA polymerase as a delta, which concentration does not increase, it means cell is not ready as for cell reproductions or DNA replications. If you also put any kind of inhibitors or that has to inhibit the activity of DNA pole, then cell cannot enter from G1 to S. So that has a DNA has a polymerase enzyme, RNA polymerase enzyme, DNA ligase enzyme, and other has enzymes, चाहे वो एक्सोन्यूक्लियेज, चाहे एंडोन्यूक्लियेज, चाहे न्यूक्लियेज, DNA ligase, 2'-isomerase, etc. That is as a non-associated as a protein that exists freely in the nucleoplasm. Okay. Second one, cytoplasm. Cytoplasm again we divide cytosol and cell organelle. Major cell organelle of the eukaryotes, when you left the nucleus, the then very first, that is the mitochondria. Second one, ER. ACR, RER, other membrane bound cell organelle, that is the Golgi, lysosome, centrosome, vacuoles, etc. In case of cytosol, there is organic and inorganic. Organic is the major concentration, that is the ribosome. Because it's a ribosome, it exists in the cell in three forms. One form, that is the dissociated, either 40S or 60S, or it is associated in 80S. Third one, the ribosome that also associates with the messenger RNA at the time of the protein synthesis, then it is known as polysomes. Not confused with the polyribosomes. It is the polysome. Messenger RNA assembles with the ribosome, then it is known as polysome, not polyribosome. Because the maximum ribosome attached with messenger RNA once a time, only one. Because molecular weight of the ribosome is much more higher than that of the messenger RNA. If has more than one ribosome attaches with the messenger RNA, then there is chances to disrupt the messenger RNA. That's why protein synthesis cannot be has to continue. Second is freely ribosomes, and third one, the ribosome attached with the membranes. RER, that is, we have written here, rough endoplasmic reticulum and a smooth endoplasmic reticulum. आपको कंफ्यूज नहीं होना है, you should not be confused, like ऐसा कि RER से राइबोसोम हट जाता है तो then चेंज हो जाएगा SCR में, ऐसा नहीं है. Because the membrane composition of these two kinds of RNA is entirely different. Rough endoplasmic reticulum is known as because it is associated with the ribosome, no. But however, it has contained the two kinds of the glycoprotein that provide the center or receptor for the attachment of the ribosome, that is ribofourin. Ribofourin was there is two kinds ribofourin. First ribofourin, second, whose molecular weight is 60,000 and 66,000 respectively. And so this ribosome attaches with the ribofourin, and ribofourin is the integral membrane protein that presents in the membrane of the rough endoplasmic reticulum. How we can select it? If I say one kind of chelators, that is the glutaraldehyde. If we also put the glutaraldehyde, then it can dissociate the ribosomes from the endoplasmic reticulum, because it has a glutaraldehyde, it has to makes the non-covalent bond with the glycophorin. Whenever glycophorin is linked with the glutaraldehyde, it does not allow the ribosome to attach. Rough endoplasmic reticulum has so many functions, later we will see, known as site for protein synthesis, but other functions, glycosylation, glycosilations, etc. Another kind of ribosome that remain present in the cell organelle. And in case of eukaryote, there is two organelle that contain the ribosome. One is mitochondria, another one is the chloroplast. Mitochondrial ribosome differs from chloroplast ribosomes. You will see later, mitochondrial ribosome that is of 55S, in spite all the kinds of eukaryotic ribosome is 80S. In chloroplast, the ribosome that is of 70S, that is similar to the prokaryotic ribosomes. Anyway. Second, protein, sugar, amino acids, nucleotides. The cell it is rich in nucleotides, because all the kinds of the nucleotides, there is two kinds, I know already, purine nucleotides, pyrimidine nucleotides. Purine है और pyrimidine. Synthesis takes place in cytosols, and as these as the nucleotides, that serve as a source of energy, particularly ATP. Anyone, can you say why has all ATP, all the nucleotides have same phosphoryl potential donor, but maximum ATP is consumed? एटीपी ही खर्च होता है, जीटीपी उतनी नहीं खर्च होती, ना के बराबर है, सीटी पे नहीं होता है, टीटी पे नहीं होता, यूटी पे नहीं होता है। अगर आप देखेंगे तो लेटर पढ़ेंगे भी, मेटाबॉलिज्म माइटोकॉन्ड्रिया पर आएगा, तो यू विल also see evolutionary in all kinds of the cell where it is prokaryotes and eukaryotes, they have evolved only one enzyme that catalyzes the energy current, that catalyzes the synthesis of ATP from ADP, or that is the energy conservation, that is known as ATP synthase. Or ATP synthase that consists of 9 polypeptide, if you have to look at from as a prokaryote to higher eukaryote also, there is similarity, and it gives the indications, prove that, provide the evidences of the evolutions, how an organism evolved from basic simple component, a simple to higher complexity. Amino acid, sugar, nucleotides, etc. Inorganic, that is the waters and ions. Ions, again, inorganic or organic. Sorry. Cations and anions. Cations, major concentration, that is the proton, sodium ion, potassium ion, calcium ion, zinc, cobalt, manganese, magnesium, etc. Major also anions, that is to maintain the pH of the SO cell, that is chloride ion, inorganic phosphate, mostly other acids like iodine, etc. Major has these two. Third one has that is the bicarbonate. Because the cell is the physiological unit that has to capture, that received has of food in monomeric as a form. That monomeric form that undergoes oxidation reduction process in mitochondria, where CO2 is produced and CO2 leave the mitochondria for the cytoplasm, or even in mitochondria also CO2 react with water, forms H2CO3, dissociates into proton plus bicarbonate. So that has this has a bicarbonate also present in the cytosol. So this is the organelle. Cell skeletal has a protein. It is also present also in prokaryotes, also in eukaryotes. In eukaryotes, there is cytoskeletal as proteins like aspartame, band as 3 protein, a question, band 3 protein, band 1, band 2, band 3, aspartame, anchoring, tubulin, microtubules. These are the cell skeletal protein that maintain the shape of the plasma membrane. Band as 3 has a protein that is present in the RBC has a membrane. And this has a RBC has a membrane. The presence of band as 3 has a proteins. Band 3 means because the proteins are isolated through the process of electrolysis. So that they are separated on that has a band that has been named as band as 3. Simultaneously band 1, band 2, band as 3. The band as 3 has a proteins present in the RBC. It has to make the RBC biconcave structure that helps the RBC to move along with the blood vessels, along with the blood as a flow inside the blood vessels, like as the blood veins and arteries, etc. This is the basic anatomy of the cell. Now we will move toward the chemistry of the plasma membrane. Any question? Anyone? Dear students, Okay. Now plasma membrane. Someone say plasma means the external covering of the cell. A cell is surrounded by a membrane known as plasma membrane, but no. We have already written here, plasma membrane is the integral as a part of the cell. It is not separate from the cell. Without plasma membrane, there is no cell. If you look at the evolutionary period, the very first organic substances were synthesized. And so these organic substances at higher temperature, they existed either in microsomes, yeah, a spheroid structure, even though proteins, thermal peptides, etc. Cell could not form unless and until the plasma membrane has were synthesized. So that plasma membrane is the sheet-like asymmetric structure and metabolically active component of the cell. Metabolically component, active. Because as the plasma membrane that also serve as barrier in between internal cellular environment and external cellular environment, some say, because it makes the barrier in between external environment and internal environment. But otherwise, plasma membrane, that is the sheet-like structure, it is asymmetric structure and it is the metabolically active. It performs various functions, including influx of the substances from external environment to intracellular environment, efflux of the synthesized component from intercellular environment to extracellular environment, that also export the substances that is synthesized in the cells, that is the cellular as a product, even metabolic product that goes out, that removed out from the cell through the plasma membrane. The plasma membrane is more permeable for the hydrophobic substances or non-polar substances. It is impermeable or selectively permeable, or partially as permeable for the polar substances. The polar substances may be ions, sodium, potassium, calcium, etc. The very first as a plasma membrane is the sheet-like asymmetric structure and it is metabolically active and regulate the inflow and outflow of the substances across the cell. In higher eukaryotes, even though in case of the prokaryotes, the membrane serve as a contain receptors. These receptors that receive the various macromolecules, even though in case of higher eukaryotes, the cell has receptor that also allow the binding of the viruses. In COVID, as you have already experienced, these are some viruses. In case of the eukaryotic cell or in case of the human, they not has enter directly because they enter through receptors, or first of all, they attaches to the membrane. In bacteriophage viruses, and you have seen bacteriophage first of all lands over the membrane of bacteria. Later one as they lyse the cell membrane through which they inject their genetic material in the protoplasm of the bacteria. So that no one has viruses, either any case of the cell, does not have to cross it directly to the plasma membrane. Now, that is the chemistry of the plasma membrane. The chemistry of the plasma membrane that was started very earlier in 19th century, 20th century, 21st century, and it is going on. But the very first indication that was given by Overton, he has to put the mustard oil in aqueous solutions that assume the spherical structure. Later one, it is called as micelles, and he called plasma membrane made up only of the lipid molecule. But in 20th century, because 20th century is known as golden era of for the biological sciences, and particularly whenever in 1933 Nolan Ruska he invented the electron microscope that is made made as a possibility for the study of the cellular as a component. In decade of 1950 onwards, 1953, DNA structure was known. 1950 onwards, the biochemistry came into existence at vigorous level. The separation technique came into existence that helped to isolate the structure of any cellular organelle. So Gordon, Grendel, Danieli and Dawson. But in case of in 1959, it was the Robertson. He gave the unit membrane concept, and he also advocated that not only the plasma membrane, but other membrane of cell organelle also that consist of lipid and protein structure. Gotter Grendel and Daniel Dawson, he also lipid bilayer and others that consist of the lipid as a bilayer, and they surrounded by the proteins. But also it was Robertson. He has put forwarded the model that consists of the lipid and protein, and he also gave the thickness of the plasma membrane that is about 75 angstrom. In that 75 angstrom, this is the protein, this is the lipid has a bilayer 2025, this is the protein. Fine. So 20 that 40 angstrom and protein has up to 35. This may 75 angstrom. ये okay, sorry. 25 है यहाँ पर 25 है, 25 है जिसमें कि 20 angstrom that is the protein and 5 angstrom it remain associated with lipid bilayer. He also gave the measurement of the 75 angstrom, and he also, whenever a unit membrane concept of Robertson came into existence, then it opened the door for molecular cell biologists for further study of the plasma membrane. But the actual chemistry of the plasma membrane came into in decade of the 1970. It was Singer and Nicholson who gave the widely accepted model of the plasma membrane that is known as fluid mosaic model. And he also told that the fluid that is made up of, that is constituted by lipid component, mostly phospholipid. I later come on that topic, and that's a protein. And he gave 2 or 3 lines. In very first line, he also told that lipid and protein are arranged in a plasma membrane in quasi-fluid manner, means semi-fluid as a manner, in which as lipids and proteins are associated in such a manner that allow the substances that has solubility either with both लिपिड के साथ जो सॉल्युबल है वो भी सेल के अंदर आएगा, प्रोटीन के साथ सॉल्युबल वो भी अंदर. So that has so he gave. And fluid, the fluid has a component that is the fluidity. Or fluidity has in last class we have introduced you, that is the, if a cell that has to contain more unsaturated phospholipids, they are more has a fluid, yeah, they are more, they have to maintain more permeability. But whenever they contain the more saturated fatty acids, so then as a solubility decreases slowly and slowly. The protein, he also told that protein is of two kinds: extrinsic protein or peripheral proteins, or integral proteins or intrinsic protein. First of all, lipid has a bilayer. A question comes from the bilayer. Bilayer doesn't have means there's two leaflet. It always has to define the presence of the ratio of the glycerophospholipids that has to contain has a two sides, polar head and non-polar acyl chain. So that has the ratio of the polar substances as well as non-polar substances that balances to both, then it is known as bilayer. However, it has to mix two leaflets, outer leaflet and inner leaflet. The outer leaflet and inner leaflet that has to contain different kind of the fatty acids, or mostly fatty acids that associates with the glycerol and phosphate, then it is called glycerophospholipids. Second kinds of the phospholipids or other kind, that is the plasmalogens and sphingolipids. Sphingolipid is of two kinds, phosphosphingolipids or non-phosphosphingolipids. एक में आपका phosphate है और दूसरे में नहीं. The glycerophospholipid where is CH2O, C double bond O, R1, and CO, CH, R2, and CHSO2O. Here attached phosphate. Later one, some attached like as a choline, CHSO2, CHSO2, and CH3, CHSO3, CH3. That is the choline. That is the phosphate. That's why it is known as phospholipids. Or the alcohol present here is glycerol. Glycerol that contains three molecules of three OH group, 1 and 2, that holds fatty acids, and as a third that has contain phosphate and choline. This is known as phosphatidylcholine. Same present is serine, ethanolamine, and inositol. In last class is phosphatidylcholine, that is also present in the outer leaflet. Outer major concentration, ऐसा नहीं कि कोई और भी नहीं है, but as the major content of the outer phospholipids, that is phosphatidylcholine. The fatty acid chain that depends upon transition temperature. Average temperature that is maintained 25 degrees centigrade, average room temperature you can see. Whenever temperature increases beyond the 25 degrees centigrade, then saturated fatty acid increases. Concentration of saturated fatty acid increases. In last class we have told particularly thermophilic bacteria that also survives at 70 degree, 80 degree or 90 degree temperature. They also contain some more saturated fatty acid along with the plasmalogens. Whenever temperature has to decrease beyond the 25, then unsaturated fatty acid increases. Or the temperature has to, at which saturation change into unsaturation, or unsaturation change into saturated fatty acids, or unsaturated fatty acid. This is known as transition temperature. Because in glaciers or हिम नदी जिसको आप कहते हैं, in that as a glaciers, the fish also survive, but upper surface of the rivers that freezes out. That has to make a shield-like a structure that has to inhibit the exchange of the respiratory gases and others also. But as beyond as the ice, the water holds and maintains 4 degrees centigrade temperature. And at 4 degrees centigrade temperature, the plasma membrane is permeable for oxygens or respiratory gases. In exchange, oxygen comes or CO2 goes out. So that this also at 4 degrees centigrade, at which also respiratory gases and other substances traverses the plasma membrane, crosses the plasma membrane due to the presence of more and more unsaturated fatty acids. अगर वहां से आप saturation कर देंगे तो वहां पर they cannot have survive. But as a temperature beyond 25 degrees centigrade, there is saturated fatty acid concentration increases in the plasma membrane. One question is also more, because as a saturated fatty acids, you can leave this distance you can see. It makes the symmetric structure. Either so makes one symmetric and the another one, it has to make distance from this to this, so that it is not parallel. It is parallel. This is also, it has to indicate the presence of unsaturated fatty acids. And whenever saturated fatty acid, one molecule, another carbon is saturated with the unsaturated fatty acid, it makes the kink. It makes the kink बनाता है. Second is phospholipids. In case of phospholipids, you can see the CHNH, this CH2O. This is contain amino acid alcohol NH, और यहाँ पर it has mostly contain the choline. Mostly contain choline or phosphate O CHSO2 CHSO2 N CH3 CH3 CH3, और यहाँ पर that is one molecule of unsaturated fatty acids. And so this has to make अगले क्वेश्चन नेट में आता है, तो यहाँ पर this is there is amide bond in a spiked C double bond O, there is R1 amide has a bond. However, in case of amino acids or proteins, the two amino acids are linked together during protein synthesis through the peptide bond. There, because enzyme is there, peptidyl transferase that synthesizes this kind of bond. In case of the polypeptide is known as peptide bond, but in this case, the amide has a bond. It is more rigid in comparison to ester has a bond. And it is also present mostly in the neural membrane. And so this sphingophospholipid also joins with the proteins, so then it is known as sphingomyelin. Or the sphingomyelin, particularly in an enzyme also present there known as sphingomyelinase, or sphingomyelinase that has to hydrolyze the choline, sphingosine, phosphate, and acetylcholine. But in autosomal recessive disorder, in case of the autorecessive disorder in African peoples, that is Jew. They have inherited an autosomal recessive disorder. They don't have an enzyme, a sphingomyelinase. Or this, so that is and the lack of the sphingomyelinase that causes depositions of the sphingomyelin over the neural membrane that retard the nerve impulses. That causes a disease already known, that is known as Niemann-Pick disease. Niemann-Pick disease, the people who suffer from the enzyme as sphingomyelinase that had nervous retardations as well as hepatosplenomegaly, where liver and spleen enlarged, and so they lost their sensation. And finally, the peoples that has inherited such kind of disorder, that is they either die within 5 years or die within the second decade, etc. Inner leaflet, that is, it contains more amines, phosphatidylserine, ethanolamine. One another has of phospholipids that is known as phosphatidyl inositol. Inositol, that is the ionic alcohol, ionic sugar. That सब पर OH है. OH, H, OH, H. And this phosphatidyl inositol that is involved during the cellular signal transductions. It gives two kinds of the second messenger. One is IP3, and second one is diacylglycerol. Diacylglycerol. Both serve as a second messenger, and so this also inositol that is concerned with, or diacylglycerol is concerned with calcium metabolisms or calcium related metabolic processes of the cells, etc. Second as a phospholipids, not as lipids are present in the plasma membrane is the glycolipids. The glycolipids, they contain the azo sugar, they don't have the phosphate, only azo sugars. If it is the galactose, then it is known as galactosphingolipids, glucosphingolipids, or gangliosides that contain oligosaccharides. That oligosaccharides, that is galactose, glucose, N-acetyl glucosamine, and further as N-acetyl galactosamines, and also further as the N-acetyl galactosamines, to which associated another as a molecule is known as N-acetyl neuraminic acids. And N-acetyl neuraminic acid, if it can present in one molecule, then it is known as GM1, and a ganglioside that contains one molecule of NANA, N-acetyl neuraminic acids, or if it contains two molecules, then it is known as GM2, N-acetyl neuraminic acids. One enzyme that always has to convert the GM2 to GM1. So that in two cases, if as a people had inherited autosomal recessive disorder of enzyme known as gangliosidase, so then as the people suffer from Tay-Sachs disease. Or in African as the peoples, particularly then in ancient ethnic African peoples, there is ratio of chances of happening of the test for peoples live as a birth peoples, that is just a 25 upon 1 out of 25 live birth. And so this as a people die as within a month, or others cannot survive. Second objective as a GM2 or GM1, if as a gangliosidase, because these gangliosides serve as a receptor as for the cholera toxins. cholera toxins. So that this cholera toxin also joins with the gangliosides and has it check that it has to allow the cell for continuous efflux of the waters and yeah, you can another point as well because it has to activate the chloride channel and aquaporin that allow the most and more influx of the water and more influx of the ions that has to cause the dehydrations as well as ionic imbalances. This is the lipid component. Another lipid component that is most notably has to that is the cholesterol. The cholesterol, or simply you can say asterol, because there's a cholesterol that was first of all isolated from the bile. That's why it is known as cholesterol. Actually, it contains the asterol because the asteroid units are this also contain OH, then as a C or CH2 26 carbon compound. So then it is known as asterol. Cholesterol is named because actually its chemical component is the asterol. This has a present in the lipid has a bilayer and its concentration varies from inner leaflet to outer leaflet and its prime functions to control the fluidity of the plasma membrane. Again, as you come, the transition temperature. As transition temperature beyond the 25 degrees centigrade onwards, the concentration of the asterol increases and as that check or regulate maximum fluidity of the cell. So that fluidity also decreases whenever asterol content increases in the plasma membrane. And just opposite, the asterol has a concentration decreases, then as a plasma membrane is more permeable in more fluid in comparison to higher concentration of asterol. Okay. Now has a proteins. Protein and as the second one is that this has to is the hydrophobic and hydrophilic. That is the polar and as a lipid acyl has a layer acyl chain that maintain the hydrophobic nature. So that is a protein is again of two types. One has been divided on the basis of their presence, or another as a kind there is solubility. Presence, that is the externally has a present, that is extrinsic or peripheral proteins, or they remain embedded along with the lipid bilayer, then it is known as integral protein. The extrinsic protein or peripheral protein that remain associated with the intrinsic proteins. The intrinsic protein which has a polar amino acids that exposes either side, to which has an extrinsic protein or peripheral as a protein attached. Whereas intrinsic as a protein that has that exists in the plasma membrane in two or more forms, or mostly there is a present in the polypeptides. And the polypeptide, if you have to look, if you have gone through the structure of the protein, the secondary structure of the protein, like alpha helix and beta pleated acids. So this protein that made that form the plasma membrane very first asymmetric. क्योंकि plasma membrane is asymmetric due to the presence of the proteins that remain embedded, that is present in the lipid bilayer, that is embedded. That's why the exist in the plasma membrane the proteins mostly exist in secondary structure. Some contain tertiary and quaternary structure, but however क्योंकि enzyme और others आपका tertiary और quaternary मिलता है, लेकिन second structure alpha helix and beta pleated structure. The alpha helix, that is a helical like structure that traverses the plasma membranes. Once a time, in last class we have introduced you, that is the monopass, bypass or multipass. once a time, twice a time. Some as the protein exist from start from here to this protein to this, and some crosses across the plasma membrane from outer leaflet to inner hexal leaflet. Whenever this has a polypeptide has so that has to remain attached has to with the lipid has to bilayer through the they contain more and more hydrophobic amino acids. चाहे आपका aliphatic amino acids, mostly aliphatic amino acids like as alanine, leucine, isoleucine, valine. Another is आपका aromatic side chains, phenylalanine, tyrosine, and tryptophan, but mostly so they contain the aliphatic side chain amino acids. That's why so they make hydrophobic. Though in case of 5 or 6, one question was asked in NET. If plasma membrane is hydrophobic or amphipathic, amphipathic means both hydrophilic as well as hydrophobic. Who maintain the plasma membrane as hydrophobic structure? So the hydrophobicity not only has to maintained has to by the lipid bilayer, but however, there's a protein, lipid has to be amino acids that remain attached with along with the lipid bilayer through the hydrophobic interactions. Integral as a membrane as a protein that exists in more than functionally it exists in more than one forms. It makes transmembrane protein. Transmembrane protein simply means a protein traverses the plasma membrane from outer leaflet to inner leaflet or from external environment to internal environment to recognize that is the transmembrane protein. All kinds of the receptors that is present on the plasma membrane is the integral membrane has a protein. Then has the channels, pump. That is it is a form of the integral membrane protein. Integral membrane में channels, protein, transporters, aquaporin, and other kinds of the protein has present in the plasma membrane that has allowed that make the plasma membrane more and more has availability for the wide functions. चाहे आपका even though during the phagocytosis, pinocytosis, these are so in case of the amoeba or other protozoans, whereas a mode of nutrition is phagocytosis and other things that is maintained as by the proteins, that is integral membrane proteins. In mitochondria, the cytochrome has this has a protein is also the integral or extrinsic protein is also there. In one has question glycero here, cardiolipin. This is also not diglycerophospholipids because it contains two molecules of glycerol and both linked with one molecule of phosphate, but however, both as glycerol has two molecules of fatty acid both. It is a cardiolipin. First of all, it was isolated from the cardiac mitochondria, मतलब heart mitochondria से निकाला गया था, and its concentration is more and more in a mitochondrial membranes. Cardiolipin. One question is important. That is the cardiolipin. It is present mostly in membranes of the mitochondria of heart or other cell other cell also.
हेलो डॉक्टर साहब।
जी सर।
आप मगर पढ़ा रहे हैं, टाइम आपका हो गया होगा।
हाँ जी सर।
हाँ हाँ, कंटिन्यू करना है, बाद में करें।
ऐसा करते सर, ऐसा सर हमें है ना 2 दिन कंटिन्यू कर दीजिए सर, अगर कोई
अच्छा, कल भी आप करेंगे?
हाँ 2-3 दिन, क्योंकि अगले मंडे को हमको गाँव जाना है सर।
ठीक है, मैं देख के आपको कल कर लेता हूँ।
हाँ तो कल परसों हमको है ना 13 तारीख को कंटिन्यू कर
ठीक,
दीजिए
मैं
सर।
रिप्लेस कर लूंगा सिस्टम।
ठीक है सर, थोड़ा शॉर्ट कर ले कल से सर, फंक्शन कल बता दे, केमिस्ट्री थोड़ा सा फंक्शन में नहीं।
अभी कंटिन्यू कर रहे हैं आप? क्या एंड करूं?
जैसे मैंने बता दी सर कहिए।
कल फिर कंटिन्यू कर दीजिए, मैं
हाँ
आपको ही
कल
शेयर कर दूंगा।
कंटिन्यू
ठीक है,
ओके सर,
थैंक यू ऑल।